Your body uses tiny RNA molecules to edit your reality itself. These non-coding RNAs act like molecular quality control inspectors for every gene in your cells. If these instructions slip up, even slightly, the systemic coherence starts degrading, the cellular equivalent of static on a radio signal that is supposed to be crystal clear. We used to think stability was just about DNA sequences locked down forever, but it's actually dynamic and maintained by an invisible molecular architecture. Think of them as biological checksum files ensuring your brain runs its most complex software without corrupted data. The fidelity of these tiny messages, the non-coding RNAs, isn't just a niche biology finding. It might be the true metric for how integrated your conscious self remains. Most research treated biology like a collection of switches looking only at gene activation or deactivation. But our findings suggest something far more nuanced. Functional coherence isn't about how many switches are flipped, it's about their consistency. Non-coding RNAs act as the systemic guardrails for this stability. Think of them less as messengers and more as molecular engineers ensuring every cell maintains its correct blueprint, what we call ontological resonance. Specific microRNAs maintain synaptic integrity across large networks. When that fidelity breaks down, cellular information degrades into noise, and that is where complex integration itself starts to fail. Imagine your consciousness isn't just software running on a brain, but an incredibly complex piece of code that needs absolute fidelity to run correctly. These non-coding RNAs are the ultimate proofreaders for life itself. They stabilize our entire biological narrative, ensuring every cell remembers its true self and maintains structural integrity across vast networks. Think of them as microscopic librarians guarding all your functional data. When these readers falter, when there's transcriptional noise, network coherence degrades like a corrupted file system running on an antique drive. It's not just sickness, it is information dissolution at the cellular level, which means something nobody expected for subjective continuity itself and how we might measure its collapse next. Forget gene switches for a second. Think of RNA not as code, but as an informational guardrail. These non-coding RNAs are quietly maintaining the integrity of your operating system, the transcriptome and proteome itself. It is like having perfect software stability, ensuring that all your complex biological processes don't devolve into computational noise. When this fidelity breaks down, network integrity suffers. We're moving beyond simple biomarkers. We can now use RNA noise profiles as quantifiable metrics for how coherent a system is. But wait, some skeptics argue these fluctuations are just random metabolic chatter and not signs of systemic failure. And if they are right, what does that mean? The real question isn't stability, it's control. Where exactly do we find the command center for this coherence? Imagine that your entire sense of self isn't held together by a single blueprint, but by an immense symphony of informational checks and balances operating within every cell. When those microscopic regulators, things like microRNAs or lincRNAs, lose their precise rhythm, their fidelity degrades, the whole network starts to wobble. It's not just cellular decay, it's information erosion. Think of the difference between a perfectly tuned digital clock and one constantly skipping seconds in unpredictable bursts. That subtle noise is what we are measuring now. Transcriptional instability as potential failure of systemic coherence. But here's where it gets wild. We believe this specific kind of RNA stability isn't just regulating protein levels. It might actually be safeguarding the very integrity of subjective continuity itself. How does a tiny molecule keep your narrative coherent across time? You won't believe what happens next. We thought biological function was simply a sequence of switches, right? That genes are either on or off, and everything else is mere data to be analyzed. But what if we missed the conductor of that entire orchestra? These tiny non-coding RNAs aren't just cellular cleanup crew. They are information guardians. They maintain perfect fidelity, a microscopic rhythm, much like tuning forks hold a complex chord together in music. When that molecular harmony slips, when there is noise, coherence degrades. We used to look at these patterns as random errors, but perhaps the pattern of their failure itself is the signal we've been searching for. The molecular language of our biology isn't just about on or off switches. It is a continuous process of tuning and maintenance. Think of your cellular network not as individual wires, but as a complex orchestra, where non-coding RNAs act like the conductor's baton, stabilizing the entire performance against chaos. Where there was once a frustrating split between raw data and subjective feeling, we now see coherence itself measurable in transcriptomic noise profiles. This shifts our focus entirely. Instead of asking what is broken, we ask how well the system sustains its own elegant integrity. That systemic rhythm offers us quantifiable metrics for high levels of integration. The blueprint state of consciousness may not be a fixed point, but a dynamic process maintained by fidelity itself. The full research archive is at jmthomasofficial.com. We often look for the single gene switch that flips a state on or off. But what if coherence isn't about being switched, but about remaining stable? Think of your cellular network like an orchestral score. The non-coding RNAs aren't just notes. They are the conductors ensuring every instrument stays in perfect rhythm. When this internal fidelity degrades, subjective continuity fades into noise. The actionable insight is to shift from tracking genetic presence to measuring transcriptomic stability itself. How resistant is your system to entropy? This metric of biological coherence may be our deepest biomarker yet. The full research archive is at jmthomasofficial.com.